Okay, thank you very much. 4 years into hybrid meetings, and we still have these issues. Okay, so my name is Natalia. Good morning to all of you. Today I'm here to present Surfaces, a software for fast quantification and visualization of biomolecular interactions. I am a PhD candidate at Professor Rafael Najmanovic's group at Université de Montréal. Um, to me, it's almost breakfast time, but it's my understanding that to you all is almost lunchtime. So I'll start by a summary of the conclusions in order to keep you interested. So basically, Surfaces is a pair residue binding evaluation that works for different structural units, for interactions between protein and ligands, and also for particular residues within chains. Our initial results show a performance that is comparable to that of some molecular dynamics based methods. Our goal with Surfaces was to build a method that was very accessible to decrease the entry barrier for those who want to evaluate protein interfaces, um, both regarding expertise and regarding access to computational power. So it is a very fast method and therefore applicable to high throughput screening. And we are using it to extensively evaluate SARS-CoV-2 spike protein and spike variants, and understand different evolutionary driving forces throughout the pandemic. Okay, so starting by the method, Surfaces is based on a very simplified approach, which is the areas in contact approach between atoms. And this was an approach introduced in the 90s and used by many other methods in our lab because it allows us to create very fast methods. And we also use a matrix of pseudo-energetic interactions based on atom types. The default matrix for surfaces is based on the 39 Sibyl atom types, and it is a matrix from another one of our softwares, Flexate, which is for ligand. But this matrix is also customizable by the user. And we basically multiply those two values, the pairwise atomic area in contact and this pairwise pseudo-energetic matrix in order to have a complementary function for some units. So we offer currently three applications. The first of them is for protein-protein evaluations, in which the user submits a complex and the chains they want to evaluate the interactions of, and therefore the units of this complementary function are residues of both those groups of chains. And as a test application, we ran it in 738 complexes of the spike protein with different antibodies. And the average it took us to run that, and the spike protein is a very large protein, was 11 CPU seconds per structure. Our second application is the protein ligand application, in which you can see the binding interaction for every protein residual and every ligand atom. And as a test application, we ran it for 131 complexes of different SARS-CoV-2 proteins and a set of small molecules. And the average to run each one of those complexes was around 4 CPU seconds. And the third application is the particular residues application, in which the user can select residues of particular interest, for example, residues that are associated with some disease or that are mutated in your experiments. And you can see all the interactions of that particular residue with every other protein residue, including intra-chain interactions. And surfaces, all those images that I'm showing are part of the surfaces visual output. So we produce both a raw interaction file as a CSV, and we also produce Python sessions with those interactions displayed visually. So once we developed the method, the initial step was to compare our results to other methods. So for that, we have used the AB bind data set of 1,100 antibody complexes. Here you can find the reference. And in this original paper, the Amy Keating group tested this particular data set and the performance of different methods to predict the effect of mutations into the binding of those antibodies and proteins. And here they modeled the mutants with using different methods and then tested different methods for those modeled mutants. Here you guys can see the performance of four different methods performing the mutants with Foldex and the performance of Rosetta performing the mutants with Rosetta both regarding persons R and regarding AUCs with different levels of confidence. And if you guys can see here the results for surfaces, we have performed the mutants the same way they did perform their mutants, because surfaces does not perform the mutants. And in order for us to have comparable structures, we had to perform the mutants the same way. So for the Foldex mutants, our performance was very similar to the top performing methods, so DFire and Foldex, both regarding persons R and the HCs. And for the Rosetta mutants, our Pearson R was slightly smaller, but our AUCs were higher than those found using Rosetta. Then we proceeded to do a validation that is more focused on the spike mutants. So here is the reference of the Sejiva evaluation, which took some mutants of spike and evaluated experimentally the effect of those mutations in binding with the receptor ACE2, and tested different methods for their ability to predict this effect of these mutations in this binding. And their conclusion was that this method here, which was a free energy perturbation evaluation based on 100 nanoseconds molecular dynamics simulations, was the best method, because they got a correlation of almost zero 0.6. And for their ability to classify mutations as stabilizing or destabilizing for this particular interaction, the phi was also almost 0.6. However, they mentioned that this is a very computationally costly method because it's based on molecular dynamic simulations, and the spike protein is a very large protein to simulate. However, for a zero 0.6 correlation, it would be worth it to use such an expensive method. We performed the mutants the same way they did in order to do this evaluation to have comparable structures, and evaluated those interactions using surfaces. And we had very similar results to those of FAP, both in our Pearson's R and our classifying ability. And the error is very similar as well. Here you guys can see in pink and blue are the surfaces results and the FAP results, which have the best tendency line visually. And we were able to evaluate those 23 mutants plus wild type in 185 CPU seconds. For our second validation, particular to spike, we wanted to compare the per residue binding evaluation, which is something that we claim we do. So for that, we took a molecular dynamics trajectory that was already published of the receptor binding motif of the Delta variant and the receptor ACE2. And we used green, which is a very well-used method to evaluate the interaction per residue. And we see here that our correlation with the results from green for this per residual energetic decomposition was 0.64. As you guys can see, we focused on a validation that was particular to spike because we have been working with spike for many years now. And we wanted to, the initial idea of developing surfaces was particular to evaluate the spike protein. The reason for that is that for the past 3 years, we have been working with the dynamics of spike and the effects of mutations on stabilizing the open conformational state or the closed conformational state. We were able to predict some of the main mutations of alpha, beta, and gamma variants in 2020 for their ability to increase the occupancy of the open state, which exposes the receptor binding motif and allows for infection, basically. However, from Delta and Omicron, here you guys can see like the radii of those variants is associated with the number of published sequences. So it's a proxy for how successful those variants were. Here we can see that the Delta and Omicron obviously were very successful variants, and they did not have an increased open state occupancy. Therefore, we realized that maybe we would not be able to predict mutations or variants anymore, because probably what was being favored at that point of the pandemic was not the open state occupancy anymore. And there are some theories about that, because that receptor binding motif is also a very important epitope. So exposing it for longer would make the spike protein more recognizable by antibodies. So we developed surfaces and we applied for that test data set that I mentioned of 738 complexes of spike. And here are different types of spike, not only wild type, but also spike proteins with mutations or new variants, all complexes that were sold experimentally and are in contact with a different set of antibodies. And here we evaluated that binding for every single residue of all those 738 structures. As you guys can see here highlighted, we see that this part, which is the receptor binding motif, is a very important epitope, as we knew already based on literature. And here highlighted, you guys can see all the positions that were mutated in any variant from alpha to BA5, which is a subclass of Omicron. So here we have the interaction of every single residue that we saw in that position inside this data set, and how well it would bind in average to that set of data of antibodies. And we also did the same evaluation for the interaction with the receptor. However, for that, we modeled those mutants, and then we modeled a conformational ensemble of those mutants in order for us to account for structural variability. We know that proteins are flexible, and for that large data set of complexes with antibodies, we already had that structural variability because this is a redundant data set. But then for our modeled variants in complex with the receptor, we had to do that conformational ensemble to have sort of the same sort of data that molecular dynamic simulations, for example, offer, which is that conformational variability of the interface. So here I show the example of the wild type structure that you guys can see here. Once again, this is the output straight from surfaces. Here is for the alpha variant and the beta variant. So we can see that for the alpha variant, for example, the mutation we see in the receptor binding motif is the N501Y, which is a mutation that increases binding with the receptor quite a lot. You guys can see it here, and also here, it's much bluer. And when we look at the average interaction with the receptor, with the antibodies, we see that this mutation actually increases interaction with the antibodies as well in average. However, this mutation appeared in the end of 2020, so prior to immunity coverage in general and prior to vaccination as well. When we look at the beta variant, we already see two more mutations in this receptor binding domain. The mutation on the K417, for example, actually decreases binding to the receptor by quite a lot. However, when we look at the interaction with the antibodies, we see that this mutation also decreases antibody recognition. And the beta variant, as well as the gamma variant, that also has a mutation in this position 417 and a mutation in position 484, were variants that appear in communities that were predicted to have been exposed a lot already to the virus and had developed some sort of immunity. We did the same type of evaluation for all variants up to BA5, the subclass of Omicron. And it is an ongoing project, so we still have to evaluate a few different things and on how to consider the effects of single mutations and how to consider the effects of all those mutations in constellations and together. But for now, what we can see from our initial results is this trade-off hypothesis in which we see that in the beginning of the pandemic, the mutations that were being selected increased transmissibility, both based on higher open state occupancy and based on higher binding to the receptor ACE2. However, with the progression of the pandemic, we see that mutations are being selected that not necessarily increase open state occupancy and binding to ACE2. Actually, some of them decrease open state occupancy and binding to ACE2. However, they increase immune escape. They decrease the interactions with antibodies or the exposure of the interface that is recognized by antibodies. Well, I would like to thank all the institutions that made this research possible and all the people that I'm very lucky to work with. Here you guys can find the preprint. This work is currently under review by bioinformatics, but the preprint is already available. And here you guys can find the tutorial if you want to use surfaces. And thank you very much for the attention.